a common protein because it besides it except the structure of protein, it have another link such as the nipple or the polysaccharide saccharide. Actually, we talk about we as the example of lipoprotein. The lipoprotein is a lipo and bound with a protein. It contains two parts, and the other three kinds of substances are the same to the lipoprotein. So those could be introduce a more stronger immune response than the common proteins. So the chemical nature is a very important I think is characteristics for the special substance. And on the other side, the molecular size is also an important aspect for the for a substance for the immune response. The higher size is more molecular weight, and the more antigenic determinants, the more complex the antigenic structure. So the relative molecular weight is positively correlated with the immune strength. You can see the molecular size go to the higher value, and the strength of the immune response is more and more stronger. And the third one is the complicated chemical structure. We can imagine that if two protein or two substance have the same or similar or closer weight, but such as A is here showed is no is only a cycle, and another one maybe is a similar weight to the cycle, but it has two special structure. Here show is a serosine, like a special structure on the surface of the molecule. So we reasonably the antigen the substance with the tyrosine is better immunogenicity than the left, because he had a very special structure on the right substance. I think it's very easy to understand this point. And more than that, the conformation and accessory of the molecule is also a very important part for the immune response capability, because we can see the diagram shows that in the three substances shared a similar structure, but it also have a distinguish for each other. we can see the left one. The left one is for label chain, lateral chain, uh, focus located on a layer, but the lateral chains is closer and closer, but the different structure on the surface of the molecular. In contrast, the middle model shows that the same distance between the lateral chains, but the cycle structure is inside and hand in the molecular rather than on the surface. So we can imagine that if they are represented different antigen, the left substance and the middle substance injected to different body. The left substance will induce the very strong immunogenicity, and the middle one is hardly to induce the response, because for the middle substance, your immune response, the immune systems is hardly to get the special structure from the middle substance, because the epitope of the middle is hiding inside. Similarly, the right one, even the cycle structure inside the molecular, but the distance is very far from each other, and so the there are enough special for recognized by the immune system. So even the middle substance and the right substance have a similar structure, but the right substance have a far distance from each other of the lateral chain. It's also easy for immune system to recognize it. So from this diagram model, we should understand that the conformation and observability is of the molecular is a very key role to for the substance to induce the immune response. And the second subtitle of this part is the foreigners. How to understand the foreigners? If the substance is a foreign substance, should induce a very strong response, strong immune response. For example, the example will be useful for understanding this part. Here showed is egg albuins. The egg albumin. The egg albumin is injected into rabbit and the fall. It's easy. I think it's very easy to imagine that after the egg albumin injected to the rabbit, it will induce a very strong. Here showed is excellent immunology, but the egg albumin injected into the form. Mostly, it is no immunogenic. Why? Because the egg albumin is from a chicken. The chicken and the form is the similar. In the other word, to expression is the the chicken and the form have a closer relationship, but the chicken has with a far relationship with the rabbit. So here shows that there is means foreigners. Mm, that means the substance which is antigenic is in one more animal species may not be antigenic in another because it is present in the tissue or fluids or of another. To uh, as, elicit immune response, a molecule must be recognized recognized as a non-self by the immune systems. The ability of immune system to recognize the self molecule is thought to occur during lymphocyte development due to exposure of immune lymphocyte to self molecule. Any, any molecule that has not been exposed to immune lymphocytes during his crucial period may be recognized later as a non-self or foreign by the immune systems. The degree of immunogenicity of a substance or the molecule is related to the degree of foreignness. Generally, greater the polygenicity distance between the two species, and greater would be the structure dissimilarity between between them, and thus greater the potential of substance to immunogenicity. For instance, borrow serum albumin is also called BSA. The BSA is not immunogenicity when injected to a cow, because the BSA is get from obtained from another kind of cow. When the BSA injected to the cow, there will no immunogenicity, but it's very strongly immunogenic when injected to the rabbit. Further, BSA exists greater immunogenicity in a chicken than in a goat, because the goat is more close relationship to cattle than chickens. So we should be understand that the relationship between the different animal is a determinate role for the foreigners for each other. This picture shows very clearly for this part. Just the sentence described the degree of immunogenicity of a substance or a molecule is related to the degree of its foreignness. And the other part of this point is the effect of a host. It also includes two parts. The part one is the genetic characteristic of the host, and the age, sex, and state of the health. Actually, the hormone and cell-mediated immune response depend on the ability of the cell to process the antigen and the present it for the recognition by the certain cell of the immune system. Maybe the different individual body have ability or the degree for the I think is different people will exist different present strength or level for the antigen to our immune systems. For example, in the maybe very simple virus forms. Well, in forms, I check the name. Wait, wait for me for seconds. Flu, yeah, flu. Influenza virus. Influenza.
Influenza virus.
Influenza virus. The influenza virus is easy to infect our respiratory tract, but in the very in the several conditions, some classmates is easy to infected, but also another classmate is not easy to infected by it. There's some difference between for the different body. Why? Because the classmate who easily easily infected by the virus, because the process of creating the antigen is slower than that of the classmate who hardly to infected by the virus. How to understand it? Because if the antigen presented events is waste more time, the virus is developing more virus. So I you should be listen very close. Don't worry. I what the example for what is the meaning or what the meaning for the example is what I want to say is different people have different ability to present the antigen, so displayed a different genetic characteristic. So here showed the is a very important characteristic for the antigen. Also, the age, sex, and the state of health is a very important role for our immune response. Maybe the boy is sorry, maybe the girl is more easily to infected by a special virus or bacteria or another something. And the old man, the age is the same. The old man is more easily to get the disease caused by the bacteria or virus, but the stronger man or stronger people or the adult man is much more have a much more stronger power to against the disease infection disease. So the factor of host is very important part for antigen for our body. Okay, we should be going to talk about another part of this, and is the classification of the antigens. From the different sites, we can have a different classification of the antigens. Here, the part one is to depend on whether lead the Th for producing the antibody. It divided into two groups. The one is thermos dependent antigen, and the another is thermos independent antigen. On the last class, we already knew the B cell to the B cell can recognize the antigen directly and get the active signal to further differentiation and then produce the antibody. But the T cell cannot recognize the antigen directly. Only the T cell, from the help of the APC, and get the fragment of the antigen. Like this, the T cell got the signal for the antigen, and furtherly to differentiated to the sensitized T cell. So actually, most of the antigen is depend on the T cell. The T cell. Here is the description of the T-dependent antigens. The T-dependent antigen do not directly simulate the production of antibody with other people or with the help of T cell. Structurally, the characterized by a few copy of many different antigenic determinants. We can see the different structure or different epitope, or we call it antigenic determinants on the surface of the molecule. different kind. We can do account for it. 1, 2, 3, 4, 5, 6. Maybe at least 6 kind of different epitope on the surface. It's very complex. This this kind of antigen must be depend on the T cell to induce the immune response. And another kind of antigen is directly stimulate the B cell to produce the antibody without the requirement for T cell help. We can see the picture also like a green layer, but on the surface, it's only one. You can see only one kind of epitope on the surface, right? The size is similar to the before, but the epitope is very simple. Only one kind, right? So it is a copy. Is a copy, one by one. Only one kind of epitope. This is very easy for our body. Is very easy to recognize. So this kind of antigen don't need the T cell to help. Also can induce the B cell to produce the antibody. So this is characterized by the same antigen, by the same antigenic determinant repeated many times. Here we showed the degree about the two kinds of antigen, the Ti antigen and the Td antigen. The Ti antigen recognized by the B cell directly and produce the antibody. But the Td antigen and it will during the different and very complex process, and at last the T cell will help the B cell to produce the antibody. Please remember, most of the antigen is belong to the Td antigen, and the little part of the antigens is belong to the Ti antigen. Maybe more than 95% antigens belong to the Td antigen. Major part of antigen belong to the Td antigen, and the Ti antigen is only include a small part of the antigens family. And on the other side, the depend on either the antigen is also depend on the relationship with the host. Here we give several special norms, such as the exogenic antigen. The exogenic antigen is the antigen from different species. Is called the exogenic antigens. For our body, for the baby, for the human. I think actually it's for the human, the microorganism and the animal immunotherapy for therapy. However, this is I think is hardly to explanation. If you have some interesting for in it, you can check uh, the keywords to the websites and know more about it. And depend on the relationship with the host, the exogenic antigen is the foreign size, foreign species. I think is another kind of species. Uh, the antigen is from the another kind of species. Is okay. For example, the toxin after some treatment, it becomes to a toxin, toxin, and the toxin injected into a horse, and the horse will be induced a very strong immune response. Reasonably, as a result, some antibody for the toxin will exist produced in the host cells, such as such as the antibody. This antibody have another name, antitoxin, antitoxin, and the antitoxin will can bind into the toxin. I think this pathway is very clear and very simple to understand, because the antibody is produced by the induction of toxin, and which produced by the host. But the antibody is produced by the host, not the human. So if the antibody injected to the human, it is also a foreign proteins, right? On this word, on the other words, the antibody, even the antibody is used for against the toxin, but as a protein, the antibody also an antigen for a human. So this means different species have different protein. The proteins from the host may be an antigen to a human being. This antigen named exogenic antigens. And another noun is anergenic antigens. The anergenic antigen is the antigens from different individuals of the same species. The simple example is the most simple example is the blood type antigen. Maybe you as a you know, or everyone know the blood type of you you already know your own blood types. Briefly, I think is a briefly introduction will be help you to understand this part. For example, the one body is the A blood type. The blood type is A, and the other one is B. Two persons stand here. The first one is the first one's blood type is A, and the second one the blood type is B. And in the first person, he has A blood type because he had the A antigen. Actually, in the first one person, he also have the antibody to the B. It also means anti-B is a antibody for against the B cell, B type cell. Similarly, the second person, which have the antibody against the A blood type cell. So we can imagine that if the first person with the is the first reason, the blood type is A, is very I think is unlikely he is lost the blood and he need blood to complement. If this if at this time the blood from the B injected to the A, what happened? The antibody the antibody B will bind to the B cell. Is useless. This diagram will show the process. The antigen of the RBC, the red how to what's the meaning of the RBC? The red blood cell, RBC. The red blood cell, the type A is it means that the red blood cell have A antigen, and the type B, the person have the RBC with B antigen, but it also have anti-A antibody. So if we use the blood of B injected to the A person, the antibody A will combine with the RBC and bind to the A antigen. As a result, the transfusion reaction will be happen. Maybe the A people will be die. It's very serious. So different type blood type cannot inject the blood for each other. And the four lungs, the autoagenic. Autoagenic is an infection and after the infection and wood and medicine used to make the cover antigen release or other antigen change or modified, which induce response for autoantigen. We can see this diagram. The RBC is a common red blood cells. Is common, and the drug is also common. But unfortunately, after the drug into the drug injected into our body, the drug will bind with the RBC. This structure maybe or has the potential to induce our body to immune response, and the B cell will produce the antibody for the drug, and the immune response happen. It's very normal. It's very normal. Abnormal is a abnormal phenomenon in the medicine. So in the medicine forward, every new drug or another medicine, and almost the drug, I think is a drug, produced and corrected after a serious experiment to check the safety for our patient. Is not safe if it induce abnormal immune response, such as the autoantigens. Is very dangerous for our person, right? So the safety of a new drug, the safety check of a new drug is a very hard or the complex work to do, and the enterprise must be brave the detailing experimental data for prove the drug is okay for our patient. The safety is very important. And another norm, I think is not very important for us. You just know that it's okay. And the second one, and the sixth is a tumor antigen. Here we motion it is to introduce this special antigen, because the tumor is developed from the normal cell. A normal cell in the special condition and stimulated and communicated, I think is more and more mutation happened in the DNA, and finally, and unfortunately, it dependent to a tumor cell, and the property of the tumor is not die. It won't going to die more and more forever. So we can see as a big structure, or a very I think is a very terrible structure for a tumor, is terrible and the structure for our body. So we can imagine that the tumor is will displace a very different from the normal cell. On the other words, one cell is a tumor cell, and the one cell is a normal cell, and put them together, we can to distinguish it, because there are not of special protein will be expressed in the tumor cell, and lead to the tumor cell displays the I think different structure on the surface of the cell, right? So those different protein from the normal cell is tumor antigen, because those antigen the normal cell have no one. So here describe it is only expressed on the tumor cell, but normal cell no expressed. Okay, another antigen-related concept is super antigen. The super antigen is the super antigen is exists no dose of super antigen is lead to active 2% to 20% of the T cell clones to initiate a very strong immune response. We can see the number 2% to 22%. It's a very, I think it's a very, how to say this? It's a very large range for the T cell, and it will induce a very strong immune response. How to understand it? We can see here. Oh, sorry. Sorry. There are the proteins that in the normal course of event, the antigen recognized occurs when the antigen is posted and post-peptide is presented with the MHC class 1 peptide complex or MHC2 peptide complex on the surface of the cell. We can see the proteogram. The APC got the antigen and digested the antigen and presented the piece of antigen, here called antigen peptide. The antigen peptide is presented by the APC on the MHC 1 class or 2 class. Here shown is 2 class. MHC 2 class. Sorry, MHC class 2. And then the antigenic peptide and MHC class 2 to form the connect I think is a title structure. So this structure recognized by the T cell TCR. This is a normal pathway of recognize of an antigen recognized by the T cell. Is a normal pathway. But how the sub-antigen recognized by the T cell? We show here. The T cell are the the TCR of the T cell, and the MHC2 class 2 of the APC closer. We can see there's no antigen peptide here, right? How the TCR and MHC class 2 closer or touch each other? Just by the super antigen. The super antigen has a layer linked between the TCR and the MHC class 2. So we can imagine that there is no antigen peptide for the T cell. The T cell have not get the special signal from the antigen. So this active of T cell is ineffective. But the T cell get the signal that it must quickly to copy and produce more T cell to immune response. But those T cell response have no direction or have no specificity. And so we can understand this after the super or antigen is inducing the T cell will be waste so much number of the T cell will be waste 2% to the 20%. So if we in many kinds of bacteria, it should be it should be expressed like this. Many the proteins from the many bacteria is very, very dangerous for human because those special proteins just like the super antigen. If our body got those proteins, those proteins will like the super antigens and bring a very, very harmful experiment for our body. So the antigen, if you is a very, I think is not a good thing for our body. Here is the show the different parts of different point with the common antigen. 1, 2, 3, 4 points. And another part of the conception is multigens. It also a substance which can active cluster of lymphocytes induce a lot of clones. We only know it is okay. And adjuvant. Adjuvant is also a substance that when mixed with the antigen and injected with it, enhance the immunogenicity of that antigen. So the mission of the adjuvant is to prolong the antigen's presence and enhance stimulatory signals and induce inglonular formation and stimulate lymphocytes proliferation and non-specifically non-specifically. Remember here, the adjuvant is the stimulate the lymphocyte proliferation is non-specificity. Non-specificity. Maybe in the final examination, I will give the question for it. You can change. You should be know the adjuvant induce the lymphocyte to proliferation is a low specificity. And the last conception is heptane and cover. Here we can see the some description is here. What is the heptane and what is the cover? We can see the diagram is very simple to understand. The heptane is a very, very small structure of chemical substance, but singly the heptane injected to the mouse, there is no immunogenic will be happen, no antibody, but the heptane is combined bound with the cover protein. The cover protein will bind with the heptin, the heptin will show the displayed as epitope, and the immunogenic will be happen, and the antibody also produced. So this diagram is a very clearly for understanding of the haptin and the covers. And the sixth and the last part is the clinical application of the antigens, such as the specific binding between the antigen and the antibody is the basis of the clinical application, such as diagnosis and identification and vaccine. Here is the example of the clinical diagnosis and the identification. Is here showed is an antigen kit, and on the in the method, briefly introduce it. And the antibody were focused on the membrane, the neutral cellular membrane, and get the sample for a patient, and put it in the left of this card, this card, and then the sample will followed from the left to the right. If the sample have the relative antigen, it will combine each other and display a line, obvious line. It means the reaction happened. Also marked with a positive result. If no the line displayed, it means negative result. So this is the basic of this kit is the antigen and antibody will bind each other specificity. Okay, at last we summarize the two classes contains is maybe 1, 2, 3, 4, six sentence, six part, seven part here. If you have some problem or some to I think is if you have some problem, you can read this sentence will be clearly about the the content of class. I want to read it, and if you want to read it, I will know if I will send it to our groups. Okay. Okay. For this class is over. Any question?
没有问题，老师。
No.
Okay. Goodbye.
谢谢老师，拜拜。
老师，Thank you，老师。
You're welcome.
See you.
Okay.
谢谢老师。
See you.